This is episode 121 of the Immunology Podcast for Microbes and Metabolism with Dr. Jayne Danska. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Jayne Danska from Sick Kids Research Institute on the podcast to talk about her research on the gut microbiome, metabolic dysfunction, and autoimmune diseases. We've also got our usual roundup of recent highlights of immunology news coming up. But first,
the American Association for Immunologists invites to submit abstracts of unpublished original research for presentation at Immunology 2026. This dynamic opportunity to showcase your research empowers you to connect, collaborate, advance your career, and access cutting-edge research. Present alongside leading immunologists in Boston, Massachusetts from April 15th to 19th. Visit immunology2026.aai.org to learn more.
all right. So we get to go on break after this.
Yeah, holiday season is here. Any plans for the holidays?
I'm going to DC for a few days just to have fun with the family down there, and my in-laws and a few other people are all just going there, and that's it.
Any family traditions around the holidays?
I mean, it's Hanukkah, which is just days and days and days of small gifts. Cook latkes at some point. That's about it.
Well, I guess on my side, I'm just going to go visit my mom for a few days and then just spend some quiet time around New Year's. Escape the city because this becomes a little war zone on New Year's Eve. Even though fireworks are not allowed in Amsterdam, that doesn't seem to deter people from throwing them in large quantities over the holidays. So that's going to be fun, but I won't know it because I won't be here. Yay.
You're going away. Where are you going?
Yeah, just to Belgium.
Okay.
A little cabin in the woods.
Well, would you like peace from your allergies?
Always. Can you offer me something for that?
Maybe.
All right. What you got?
So we got a paper in Science Translational Medicine called A Vaccine Targeting Human IgE Induces Long-Term Protection Against Anaphylaxis in Humanized Mice. First authors are Eva Condi and Emma Lamana. Last author is Laurent Reber. Came out the 3rd of December. And they worked on a vaccine to human IgE that then reduced allergy. They use humanized mice as a test bed, which is a decent system, right? I'm looking at anaphylaxis. So the idea is, so one of the key parts of this is they made the antibody not just to bind IgE, that doesn't work, but to hold it in this closed and active system that's based on the drug that's already available, because there's a monoclonal antibody for IgE already. And so trying to generate something that has the same structure-function relationship as an antibody itself. So that was kind of the part of this. So, because the monoclonal is super expensive, as you would expect, right? So they looked at the crystal structure to understand how things worked. They made a mutation in a protein, an IgE, that would force that conformational change to happen that the antibody tries to generate, to kind of then create an antigen that had the right shape to play with, right? And then they created a antibody that had a CRM carrier protein, which I don't really know how that works. It's a carrier protein that's attached to the fragment of the IgE they're trying to generate, so making what's called an IgE-K antibody that's on the C3, C4 fragment. And then they made that, and they gave it to mice. And it worked. No, I'm done. No. So they figure out where the antibody binds, which is near a glycosylation site that's important. And they know if they get rid of that glycosylation site, it alters the effect in humans and in mice. They have to generate these humanized mice and then test it. And then they give the mices various antigens um, for to test anaphylaxis reactions, right? So they something called passive system anaphylaxis, which is a model of severe reactions observed with grade 2B3 anaphylaxis. These usually requires medical intervention. So they do anti-NP human IgE intravenously, followed by the next day intravenous injection of this NP BSA. NP is the antigen they're using here. And they do that. And they also do a cutaneous system as well, using the same. NP is anti-nitrophenol, which is on the back of the skin. So they do a skin allergy, they do an anaphylaxis allergy. And then they look at all the things you look at, right? So they look at both the immune response, they look at extra vasodilation, leaky gut, Fitzy dextran, right? Gavage, see that leak out. They, look at third spacing, which is when fluid leaves the blood vessels into the tissue. They look at, mast cell activation and show that the antibodies really work, that there's no hypothermia, so on and so forth. And then they look at how long lasting it is, and they show that it lasts for a year after the one vaccination. They do a booster one time through, I think. They have a 14-day booster, but then they're done. And then IgE is important for preventing, parasitic infections. So they did a parasite against hemolytic Strongyloides radi, and it didn't affect the ability to clear that. The type 2 immunity was intact. And so I really appreciated they did that last little bit, like, hey, are you screwing up the immune system with this vaccination? Well, basically what it looks like it does is it pulls the IgE levels way down in response to any antigen, whether it's a mild allergy or a severe allergy, and it keeps the IgE in not gone. It's not a soak, but it keeps it in the closed inactive state. And it circulates for up to a year afterwards with just the initial course of doses. An adoptive transfer of sera works as well, right? So they're showing it still has an effect. Obviously, it's not persistent in the same way, right? But that you're getting an immune response here. So it was a pretty well done paper. The humanized mouse, I think, is a really good first step, right? If you need to do something before you shove this in people.
Yeah.
I was like, okay, legit.
Jason approved.
Yeah, that's where I'm at. Look, it's going to be really great instead of just a monoclonal.
Yeah. I guess, yeah, my question was going to be what happens with the normal use of IgE. But I guess I'm thinking about the difference when you have allergy or when you're like atopic or whatever, you have this huge amounts of IgE, right? It's
Yeah,
so it really that only have that really is associated to some kind of allergic disease. So I guess in a way, you for normal functioning, you don't need a lot of IgE for what IgE is supposed to do. So I guess that's what they see here. That even if they're I guess they show that the amounts of IgE in the serum reduce dramatically. Um, then it's not necessary. It's really a more of a pathogenic thing to have too much IgE in your plasma.
exactly. That's really what they showed. Is it like you can just knock it down and it's fine because you're not supposed to have that much normally.
Yeah, yeah, that's pretty cool. Are they? So this is all in mice.
This is only in
Um,
mice so far. And obviously, the next step is human, but they don't have any human bridging data.
but these are humanized mice. Are they making human IgE or is it
Yes,
mouse?
it's human IgE.
Okay, so they can use the same things they're targeting, the same epitopes they're targeting in this vaccine should be also applicable for a human vaccination.
Yes.
Okay. That's very interesting, especially for people with severe allergies. This could be an interesting therapeutic proposal. All right. So, given that we are in the allergy field, I also have an allergy story for you.
What? Copycat.
I know, I know. This comes from my friends, also at Science Immunology in this case, and it's a story that looks into the origins of allergy in certain children that develop allergies. Statistically speaking, or kind of epidemiologically speaking, there is an increased risk of having allergies or having asthma, actually, if your parents also have predispositions. They also have asthma, or they have severe respiratory allergies. So always the question is, what are the mechanisms for this correlation? And of course, an obvious mechanism, the first thing you think is, well, it's a genetic predisposition, and that is important. But in this paper, they show that there's more circumstantial things that happen around the perinatal phase that actually also increase the risk of developing asthma if you have an asthmatic parent, and that's what this paper is all about. So the paper is called Maternal Allergy and Neonatal RSV Infection Synergized via FcRn-mediated allergen uptake to promote the development of asthma in early life. There's three first authors: Elizabeth DeLeeuw, Josephine Eustason, and Cedric Bostales. From corresponding authors Hamida Hamad and Bart Lambrecht at the VIB Institute and the University of Ghent in Belgium, so here are neighbors. And as I mentioned, this paper is looking around. The study tries to understand the connections between asthmatic parents and asthmatic children. It starts with looking at a very large data set of children born in Denmark. They have this huge data set in which they have been following children throughout their lifetimes. And there's a lot of examples in which the children become infected with the human respiratory syncytial virus, RSV. And this is a fairly common infection in children, but it can be very dangerous. Sometimes children are hospitalized. And so this data set is really documenting children that become hospitalized. And they show that the risk of becoming asthmatic or developing allergic rhinitis is increased is associated with having presented with RSV infection as a child. And of course, in this case, it's only kind of severe enough that you get hospitalized for it. But in principle, they see that there is this strong correlation with having been infected with this respiratory virus in your early age and then developing asthma. And I think this also is kind of already established in other. I think this is already established that there is this correlation in children. So they find this so they look at this in this data set. They really show in this data set that there's this synergy between having your mother having maternal asthma and being hospitalized with RSV. It gives you the highest cumulative chance of becoming asthmatic yourself, and it's much higher than just having your parent being asthmatic. So there's something about this event that seems to correlate, that seems to increase your risk of really developing this allergic response. So what they do is they use a mouse model in which they infect the mice with the different virus that is like a mouse specific strain that orthoneurovirus murus that it imitates the RSV infection. And they show that, kind of in a nutshell, that in children, that being infected with this virus will, of course, activate your immune system, right? So you end up having, in particular, they show that the dendritic cells become certain population of dendritic cells, CDC2s particularly, but also CDC1s, they start upregulating activation markers. And in particular, so you have the classic CD80, CD86, CD40, and CDC1s, but in CDC2s, they start expressing a lot of Fc gamma receptors, CD64. And this seems to be a key connection with this increased risk of asthma development. Because what they suggest, so they're showing this, and they do a lot of experiments, and the conclusion that they are reached is that dames that have, so they induce allergy in these dames that, or they transfer antibodies against allergens through the breast milk, or they add it, or they give them to the pups. If you combine having access to these allergen-specific antibodies through, which would presumably come from your allergic mother, and if you combine that with an event, an immunology active event in your respiratory system, which is the RSV infection that activates your dendritic cells and makes them increase their expression of Fc gamma receptor, you end up capturing these antibodies that will kind of initiate this loss of tolerance to these allergens, because you have the antibodies from your mother, and on top of that you have a activated immune system, and then instead of developing tolerance, which would be the desired consequence of having these antibodies in your mother's being transmitted from your mother, you develop Th2 response, or you being the mice, right? So I thought it was really, really interesting how they showed this mechanistic connection between these two seemingly unrelated events and the consequence, the epidemiological consequence that they have down the line, which is an increased rate of asthma development. So, of course, they show this. This explains the effects that they see in. Well, this could help explain the correlations between mothers and children being asthmatic. Of course, while the fathers that's not going to explain the contribution of fathers, which in human populations you also see it. So that might be more due to the genetic predisposition, to the predisposition to being atopic, which is have a higher chance of developing antibodies in your early age. But this RSV infection seems to be really the key that gets your immune system out of whack. And if you have these antibodies that maybe were not going to be problematic, they will increase the chances that these antigens become presented, and you end up developing an immune response against them. What's interesting is that they say that they show that if you reduce the immune response to the RSV, if you do immunotherapies to reduce the infection of RSV, or through vaccinating the mothers or providing monoclonal antibodies around the time of birth, then you can help the children, or you can help the pup, in the mouse situation, not have such an increased immune activation. And this actually helps reduce the chance of developing asthma down the line. And it's actually also a possibility nowadays, a therapeutic option for children that can also be treated with monoclonal antibodies. But I think so far it's not clear whether that in humans has shown an actual improvement or a reduction in the development of asthma. But I think it would be very interesting to study, because if you can only help children, not becoming sick of RSV, but also help their chances down the line of not developing asthma, that seems like a very cool intervention to do. So very nice paper, very nice mechanistic insight. Of course, the human situation is more complicated, but I think this provides a mechanism for this connection.
So RSV strikes again, huh?
Yeah, RSV is, it's like EVV. You just keep getting all these undesired effects. So we should probably not get it if we can prevent it. It's not a harmless infection. It wasn't obvious already.
Well, definitely hurts the babies.
Yeah, yeah, for sure.
But I think showing that there's linkages for other people to get it too is important, right?
Yeah. Yeah, so I mean, this is all about early onset of asthma. So this is mostly about baby and young children.
Right.
But this infection that you get as a baby then is going to increase your chances of becoming asthmatic down the line. So I think that's very interesting. Very interesting to keep that in mind. So many factors contribute to
Well, I
your
think that's the
immune.
point. It's one thing, I get the infection once, but now what if I actually, have a sequela from not
Yeah.
being vaccinated, right?
Yeah. 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 Get the children vaccinated, people.
Well, let's talk about the lungs some more as a segue, I guess.
Ooh, okay. My favorite breathing tool.
So this is in science. It's macrophage MR1 antigen presentation promotes mate cell immunity and lung microbiota modulation. Huh. First author is Jayne Danska, co-first author is Yuting Yan, last author is Hamish E.G. McWilliams, and other last author is Jose Villadongos. So, mate cells are mucosal associated variant T cells, right? And they hang out and they're part of the weird immune system we have with the mucosa. So, I am trying to understand why this paper is so cool to be in science, and it took me all of reading it, and I still don't understand. But what they did, and what I can go over, is that so we know they're important for tissue homeostasis and antimicrobial immunity, and they're invariant, but the cells that present and the antigen that is really important for them is vitamin B derived antigens. And this is important to know. Vitamin B is a vitamin produced only by bacteria. So anytime if you are having vitamin B in your life, you got it from a bacteria, which means you add a little bit of poop in your diet, which is fine. Everyone does that all the time. Trace amounts, right? Including vegans who, get it from their vegetables or anything else. So there's this vitamin B antigen we know is important. But then the cells that present the MH class 1 related protein 1, MR1, so that present the vitamin B antigen to activate the mate cells around them. So this is what they basically figured out. And they figured it out pretty quickly. And then I'm trying to figure out where and maybe it's just they figured it out and that's what the big deal is. So they did a TD tomato system with the MR1 gene that are heterozygotes and then tried to figure out where they were. And so they identified various cell compartments that had them, but this is a pretty specific protein. They looked at hematopoietic cells. The only one that had low level, consistent levels were alpha beta cells in almost all tissues, and that it's known to sustain mate. But then the myeloid cells, they found them in macrophages, in the lungs, the pleural cavity, and the percutaneous tissue. And I will say, I don't often call out writing in papers, but this paper the peritoneal cavity. They use a bunch of non-conventional acronyms all over the place and makes it very hard to follow the whole thing because it's just acronym. And it took me, I had to read it twice as a result. So peritoneal cavity is PER big C.
Oh.
I'm like, it's the per C, and then there's and they shorten macrophages to the phage symbol M phage everywhere, and then IM phage. And so long and short, the large peritoneal macrophages, LPM, and alveolar macrophages of the peritoneal cavity and lungs were the big ones. And they're not typically replaced from the embryonic yolk sac, although if you deplete them, they do get replaced. And they show that these are the cells that produce the MR1. Then they tie it to the microbiome. So they show that the captures exogenous ligands, so they can use a chemical analog, MR1 antigen analog that's labeled, and they can see it integrate. Cool. They show that the tissue environment and the microbiota are bidirectionally important. So germ-free mice don't present as much. Duh. And that knocking this out screws up the microbiota some. Okay, they're tied together. It's a microbial sensor for vitamin B antigen. Duh. Right? So they link that in. They then demonstrate that some of the genes involved, and they're bacterial sensing genes like the MID88 related genes, the TLR related genes, right? And that's really important for them to produce the MR1 gene expression. Okay. And then they go and shows that if you present MR1 presentation by macrophages drives MATE1 cell protection against infection, right? So if you get rid of this and then give them nasty bacteria, in this case, Legionella longbeachii, which means it's probably found in Long Beach, California, or Salmonella, it does not go well, right? So
Okay.
the mate function. So like, I think the big deal is they isolated it to these macrophages, and not all macrophages, but these macrophages in these tissues are really the ones that drive this, and it's important for presenting this. And then all the microbiome and everything else being important, but they identified the cell type that produces that presents this antigen to mate cells. So they found the other thing that the mate cells mate with. Ha ha ha.
Don't be so enthusiastic about it, Jason. You're just
Well, I was
blowing me
going in
away.
super excited, and I'm like, okay, really cool. And I was waiting for that second punch. Like, usually there's a 1-2 punch in the paper. And they really just like, hey, like, it's these cells, the microbiome's important. Yeah, I know. Yeah. Like they check some of the boxes that I would expect to be checked.
Yeah.
Right? But also not super exciting after they figured out that was expressing the antigen.
All right. Well, but I guess that if they didn't know who was interacting with the mate cells in the first place, this is a big deal. If there's really a subpopulation of macrophages that is doing this, I think it's good to know that that's how it is. So, all right. Well, thank you for sharing. Very interesting. And you know what's more interesting?
Right.
People that get miraculously cured of HIV and AIDS. Isn't that cool?
Yeah.
So I have my second paper of today is called Sustained HIV-1 Remission After Heterozygous CCR5 Delta 32 Stem Cell Transplantation. And yeah, as the title already indicates, this is basically a case report of a patient that is for all intents and purposes cured of his HIV infection after receiving a stem cell transplantation because of leukemia. But there's a twist to this that I think is very interesting. So this it was published in Nature, published on the 1st of December. First author, Christian Gabler, and Samad Kaur from the group of Olaf Pennack at the Charité University Hospital in Berlin. So this, as I mentioned, this is a case report of a 60-year-old patient that 10 years ago, almost, received a bone marrow transplantation. And to understand the relevance of this, let's give a little bit of a history class here, which is there's only been in the history of medicine a handful of people that have been considered cured of HIV/AIDS. The most famous and the first one was a famous Berlin patient, Timothy Ray Brown, who what happened is that this person was infected with HIV, but on top of that, you're really lucky he was affected with acute myeloid leukemia. And for that, he received a bone marrow transplantation. And he received it from a donor that had a very particular mutation known as CCR5 delta 32, in which the co-receptor of HIV, CCR5, this chemokine receptor that is, very well expressed on certain CD4 T cells, and that together with CD4 is necessary for HIV internalization and infection of T cells. He received this transplant, and eventually after picking up the transplant and starting expressing the immune cells from the hematopoietic cells from these donor, he discontinued the retroviral treatment and remained HIV free for many years until his death of a recurrent AML. But basically, that was the first case, the first published instance of a person not becoming HIV negative after being infected. And there's been a few other patients, just a handful of other patients, but they have always had something in common, which is that the bone marrow that they receive is homozygous for this CCR delta 32 mutation. So both copies of the gene. So they don't have a full-length version CCR5 molecule on their surface that HIV can use to enter the cells. And it was always really thought that this was a requisite for kind of mediating this quote-unquote cure for HIV for patients that were unlucky enough to need a bone marrow transplantation on top of their HIV treatment. And this kind of became established thought that, you really needed to have this. So the problem is that CCR5 delta 32 homozygous patients or like donors are very rare. They're about 1% of the Caucasian population and even less common in other ethnicities, almost non-existent in other races. So very non-practical to count on these people. And also bone marrow transplantations are very risky, so you would not just give them as a treatment for HIV. So, but in this case, so basically what is interesting about this case study is that this person has become HIV negative, but after receiving a heterozygous CCR5 delta 32 bone marrow. So it's not completely absent, but it's just a lower amount of wild type CCR5. And he himself, the patient himself, is also heterozygous. When you look at the kind of the clinical history of this patient, he already had kind of a milder HIV infection and AIDS for a long time. He had relatively low viral titers, relatively almost healthy CD4 levels. And then at some point he got worse and he started going to antiretroviral therapy. And then when he was diagnosed with I forgot which kind of leukemia, AML as well, then he received this allotransplantation. And because they couldn't find a donor with a homozygous for CCR5 delta 32, they just gave him a donor that was heterozygous. But so he was conditioned, he received the transplantation, he was treated with radiotherapy and chemotherapy to remove his original bone marrow. He was replaced. And then eventually he was not put on retroviral therapy, but he did not develop viremia, and he did not have any problems with his T cells. And for many years, they have been doing very, very sensitive HIV genome analyses, and it shows that it's basically undetectable. His T cells, they do in vitro studies, so in vitro cultures, the T cells he has, they are still susceptible for infection because they still express some CCR, kind of wild-type CCR, so they're not refractory to infection. So that is not the mechanism that is here. So people are scratching their head. It's like, how did this person become HIV negative? And there's probably a kind of many things. So there's other studies in which people have become, for a time after bone marrow transplantation, they had undetectable levels for a long time, but then they relapse. So there seems to be some kind of delay. And from all the studies that they did, I think the most interesting is they show, first of all, that this HIV, which is important, with time, it happens that this HIV-1 strain has a tendency to switch from CCR5 co-receptor to CXCR4 co-receptor. And this has to do with certain mutations in in the viral proteins that allow this change, and certain level of pressure for CCR5 and kind of to reduce expression of CCR5. You lose cells that express CCR5 because they die. So there, it is known that HIV will kind of slightly change the tropism to CXCR4. This is not the case for this patient. This was a virus that was very dependent on CCR5. And what they show is that probably, so that is not what's happening. But I think what I found most interesting is that at the time of transplantation, they had a very strong ADCC activity, so antibody-dependent cytotoxicity. And there's also the effects of the graft versus, in this case, kind of viremia effect from the donor cells that they are very immunologically competent, and they can attack cells that are carrying the virus. And what they seems to have happened is that very early after transplantation, there was this kind of burst in ADCC activity in which the viremia declined very strongly and never recovered. So there was kind of this reset situation in which, because of the transplantation, because of the certain mismatches also probably between the donor and the patient, that was kind of mediated a little of a graft versus host situation in which the original infected cells from the patient were attacked, and the new cells did not get infected, so they were not carrying the virus. In principle, just resulted in this patient not having any replication-capable viral genome that is basically present in his body. So it's very interesting because it shows that the absence of CCR or wild-type CCR5 is not the only mechanism, and that maybe you can help this immune reset that happens around the time of transplantation to mediate, to kind of force this viremia, this specific depletion of HIV positive cells and of HIV infected by the graft cells from the donor. And I thought it was very interesting because it really provides, maybe you can make more advantage, and you can use heterozygous patients to treat patient to provide a more curative option for those who need a bone marrow transplantation and have HIV at the same time.
I mean, that's super interesting.
Yeah. Yeah, I thought it was really cool. And and this is like literally a handful of people that have.
Yeah, it's all these similar types of mutations and stuff, right? But that's the story, is that it prevents it from getting in and functioning, right?
Yeah, but not in this case, right? This was
No.
a story until now. That was the assumed wisdom that you had to have, that your new cells had to be refractory to HIV in order for this thing to work. But this patient shows that that's not necessarily. Of course, it's better if you have a homozygous delta 32 mutation. Sure, you're good, but that's not necessarily the only way.
But you have to have at least heterozygous. You can't get it.
Because it also has been shown that heterozygous patients do have better outcomes and better control. Like they can control the virus in some patients to like better than people that have that are wild type homozygous. So that already gives this person a head start. So that's for sure.
Yeah. Well, Brenda.
Jason.
We're going to be speaking with Dr. Jayne Danska at the Sick Kids Research Institute in just a moment. Before we get to that, are you studying T cells? Check out the new Nature Reviews Immunology T cell nomenclature wall chart, created in partnership with STEMCELL Technologies and expert immunologists. This free resource offers a modern modular framework for classifying T cells by lineage, function, migration, differentiation, and antigen context. Bring clarity and consistency to your T cell research. Get your free copy today at stemcell.com/T-cell-wall chart.
That's actually pretty cool because I shared that paper with my group. So I wanna, dear stem cell, can I have a poster, please? Thank you very much. I'll be waiting for it. For the second part of our episode today, we are delighted to have with us Professor Jayne Danska. She's senior scientist at the Hospital for Sick Children Research Institute and professor at the University of Toronto in Canada. And her lab works on the genetic and environmental mechanisms and causes for autoimmune disease, particularly type 1 diabetes. Very interesting research around those profiles. So I think also microbiome. I think Jace is going to be very excited to start our conversation. Thank you so much, Professor Danska, for joining the Immunology Podcast.
Thank you for having me.
Thank you for being here. All right. Microbiome. I'm just going to jump into it since it seems to be an area in your research you've picked up on more recently. You have some interesting work on the role of the microbiome in diabetes. So I was wondering if you could talk about that first, especially in relation to type 1 diabetes, because I think it's super fascinating since type 1 diabetes, this prototypical autoimmune condition, but now the microbiome has a role as you've been describing, and what the heck's going on and why is the microbiome awesome? I'm biased.
Yeah. Great questions. Thank you, Jason. I think the trigger for our group and many others looking at the intestinal community of organisms or the microbiome in the context of autoimmune disease is the recognition that the frequency of a variety of autoimmune diseases, including type 1 diabetes, has increased substantially in multiple countries around the world, let's say since the end of the Second World War, over the last 70 years. And when you see changes in the epidemiology of diseases, their frequency, the age at which they present over a short period of time in a population, in multiple populations across the world, that is telling us that these are not due to human genes, because our genes don't change that fast, right? So it focuses our attention on environmental features that have changed probably very dramatically since the end of the Second World War in many places on the planet. Why the microbiome? Because it is a population of really fascinating organisms, and we're just scratching the surface on what they are and what they can produce. But we know from lots of work in the past that they are a critical interface between the host, the human, and our environment. So when we don't really know what these environmental features might be that are associated with increases in frequency, for example, or increases in incidence of type 1 diabetes, we're looking to this critical interface between the host and their environment for potential clues. But sort of the big picture of how the intestinal microbiome sort of lands in the middle of groups of scientists, including myself, who for years really focused almost exclusively on the human genetic underpinnings of diabetes risk.
I'm going to say something that's very obvious, but I hope you can help us elaborate more on this. It is really difficult to study these things in human populations, right? It's very hard to understand the influence of things like the microbiome. You also have some work done on sex differences. How do you go about looking at these data in humans? Do you do modeling in mice or in research animals to try to understand the mechanisms and the causality with the correlations that you see? How do we find a way to pull apart this complexity and get meaningful signals?
Sure, these are key considerations that really have directed our priorities and our work in this field. So, to your important points, we want to ground what we're looking for in the context of humans, and in our case, we're particularly interested in little humans in early life, because this is where probably the precursors of type 1 diabetes risk really are generated. And in addition to the evidence that the incidence of this disease is increasing, the age at which it presents, or so-called age of onset, is younger than it was, for example, in the 1950s, 60s, 70s, where age at onset was collected. So we're interested in early life. How do we collect that kind of information in humans? I think the key in the human studies has been really fabulous work from consortia of research labs that have managed to convince and recruit usually pregnant women who are interested in this problem into so-called longitudinal studies. And the key features of of studies that allow us to begin to probe these kinds of complex questions about diabetes risk and the microbiome are those that are able to bring in the children very early in their lives, often, as I mentioned, since they're not fully sentient yet, with recruiting their mothers and other caregivers, and then with research designs that allow for the longitudinal repeat sampling of fecal samples, blood samples, sometimes breast milk samples, as well as a rich bevy of clinical metadata about the children, and in some cases also the parents, depending on the design of the study, and to give us the opportunity to do repeat measurements, either of the composition of the fecal microbiome in those kids. In our case, we've also been quite interested in their immune responses against the microbes in their gut. But whatever the evaluation platforms are, whatever the omics that different groups are doing, having these kinds of beautifully designed studies, which are reasonably well powered and have longitudinal repeat measures and that consistency of rich clinical metadata, that's really been key to the clinical or to the human-based studies. And there are studies based in Europe, in Scandinavia, in Germany, as well as in the United States and other places. And I think having this geographical diversity has also been a very important feature. And we can circle back to that if you want, because I think that there are health equity issues that are embedded in any kind of research problem, and they certainly are true for the problems we're talking about today. So that's the kind of the human study pieces. These have been really critical, these longitudinal, often with a nested case and control design, by which I mean where within that same population, that same cohort, you've got children who do progress to type 1 diabetes, or else clear biomarkers indicating that they're moving toward type 1 diabetes, and also matched controls inside the same study being evaluated in the same way that do not progress, right? So this nested case control design and longitudinal sampling, I think these have been key for the design of human studies. For the causality that you allude to, we all agree. As hard as one works to do highly resolving studies, statistically robust work in human studies, at the end of the day, absent an interventional clinical trial, it's very difficult to show cause and effect. And even in interventional clinical trials, it can be quite difficult to show what is the mechanism by which the intervention is working. So to try to address those issues, which are key, our group also uses some mouse modeling as a companion to our human studies. And so, for example, we've done a large study, which we're finishing the data analysis for right now, where we isolated hundreds of bacterial strains from fecal samples of children in one of these longitudinal nested case control studies that I mentioned earlier. And we characterized all of those organisms. So we sequenced all of those organisms. And then by being able to associate those organisms either with the children who did progress toward type 1 diabetes biomarkers, these were actually antibodies in their blood that recognize their own beta cell antigens, right? So-called islet autoantibodies. So kids who did progress to islet autoantibodies versus controls who did not. We were able to take the strains that we isolated from these children and determine of those which kind of constitutes the core microbiota, in other words, organisms that we saw in both kinds of kids, and which of them did we only find in children who did progress to display islet autoantibodies versus their companions in the cohort that did not. So we had all of this information, and then we designed consortia of microbes, consortia of bacteria. These consortia were about 30 bacteria per consortia that were composed of the core, that is the core organisms that we saw in all of the kids, and then either a group of organisms that were specific to the islet autoantibody seroconverter children versus the non-converter children. The kids in my lab described this far better as pasta with two sauce, right? So we have the pasta are the core organisms that we saw in all of the kids. And then we have a tomato sauce and a pesto sauce, right? Depending on organisms that we only saw were exclusive to kids who either did or didn't progress. And we've taken these defined consortia. We could grow up large amounts of the organisms, wash them, sequence them, all of those good things, combine them together, and then freeze hundreds of replicate samples. And then we use those samples to colonize formerly completely germ-free or sterile mice. At just when they were weaned from their mums, we colonized them with one of the two types of consortia that I've mentioned, and then we watched for a variety of immunological parameters, metabolic parameters, and so forth, what happened to these mice. And the mouse strain we chose is one that is inbred and has a high frequency of spontaneous autoimmune diabetes. So in that kind of study, the idea was to take bacteria that arise from these children. We weren't testing mouse bacteria, we were testing the children's bacteria, and to compose a small sort of mini microbiome that was reproducible and that represented these two different states of children who did or didn't progress, and then use the mice as the readout. So that's just one approach to trying to address the causality question you've raised.
So in terms of that, though, so mice are, corpophagic and not humans with human immune systems. Do you think that it's really the bacteria driving it, or do you think there's genetics in some humans where HLA or TCR variant or even epigenetics, right, that then sculpt the microbiome, and then those same things are what make you prone to type 1 diabetes? So I'm saying there's some immune thing that makes you pick some bugs that stick around, and those same immune effects or related parts with the same kind of host factor drives the autoimmunity for type 1 diabetes.
Sure. There's massive evidence for strong host genetic effects in type 1 diabetes, and you've seized upon the golden piece, of course, which is HLA polymorphisms, Jason, and we've known for decades, long before the advent of genome-wide association studies, we knew from old school serology that HLA variants were very highly associated with type 1 diabetes risk. So there's no doubt about that. Strikingly, there has not yet been a strong signal that the HLA variants that any given, let's just say, kid in one of these longitudinal studies, right, that these children have actually sculpt the composition of their microbiota. That is, that you can see affiliations between particular taxonomic distributions of bacteria and whether you're HLA-DR3, HLA-DR4, or some other haplotype. And I think a lot of us expected we might see that, but at least given the data that's available now, we don't yet see that one-to-one relationship. And I guess it is likely that a variety of different genetic variants that any given human carries affects in some way the composition and or the functionality of these microbial communities. It's just that we don't have a strong handle yet on direct associations between particular host genetic variants and particular compositions of the microbiome. So they may still be there, but they haven't been robust enough to be identified yet. And that doesn't mean that there aren't important associations. For example, I mentioned earlier that we've taken a sort of the immunologist's eye view toward the microbiome in the context of type 1 diabetes to ask, what does the immune system actually see? Yeah, you've got hundreds of different bacterial types in the intestine of children, but what does their immune system respond to? And we've done that by tracking antibodies in the blood that can recognize these particular bacteria. And there we have seen very robust associations between particular HLA haplotypes and antibody responses against particular types of bacteria in the gut. Very, very strong associations, which probably are telling us that these antibody responses are T lymphocyte dependent, right? Because it's the T lymphocytes whose receptor repertoire and so forth is sculpted to recognize HLA molecules cradling pieces of proteins. So there we definitely see strong relationships between host genetics. In fact, the key host genetic feature for type 1 diabetes risk, HLA class 2 haplotypes, and the nature of the antibody responses they make against their intestinal microbes. So that's one example where your hypothesis or your predicted hypothesis has been borne out.
So, but I missed something. You before Jason questions, you mentioned that you had these strains that you isolated from the patients, either that had or had not gone to develop type 1 diabetes. But I'm missing, what are the results in the mice? Do you see that they are causing, like the mice that receive the microbiome from affected children have a higher chance of spontaneously developing diabetes in this model that you studied?
Yes, so we're readying these data for publication right now, and what we have observed in this particular model, the germ-free version of this mouse strain, which is called the non-obese diabetic mouse or NOD mouse.
Yeah,
germ-free NOD mice actually progress to autoimmune diabetes at quite a high frequency. So in the model, you don't require a microbiome to read out the autoimmune diabetes. So really the signal that we were looking for was protection.
Okay.
Right. So in our study, we see quite a robust, statistically significant difference in the incidence of type 1 diabetes. It's really an autoimmune diabetes. It's not really type 1. It's the animal version of this autoimmune diabetes. We see a much higher frequency either in germ-free animals or animals that were colonized with the bacterial consortia from seroconverted children, whereas the consortia that we put in from children who did not seroconvert, at least in their first 3 years of life, was highly protective against type 1 diabetes. Mm-hmm. So we have a very strong signal there that suggests that at least in that model where we've taken the human microbes and put them in a mice, the microbe consortia that we constructed from children who did not seroconvert were highly protective against autoimmune diabetes in the model. So that would fulfill the causality idea in the context of that model. And of course, we've tracked very, very carefully the engraftment, the colonization of each of the 30 organisms in the mice over time. We've done quite an extensive evaluation of their immune compartments, both in the gut and proximal to the islets and so forth. So we have a lot of data. We have metabolomics data, metabolites in the fecal samples as well as metabolites in the serum of the mice. So we have a lot of different kinds of evidence that we're attempting to integrate to try to understand how do these organisms coming from the non-seroconverted kids protect against autoimmune diabetes, whereas very similar compositions of bacteria that came from the seroconverter children did not protect.
Interesting. So even though the composition's similar, there's a different chain. Do you think it's bacteria related or host? Like, are you seeing different glycosylation, different, RNA-seq profiles of what they're pumping out?
So you mean the bacteria?
Yeah.
The pasta is similar, right? Because the pasta was selected to be the same, the core community. And then there are a handful of bacteria that were introduced to those consortia, which we only saw in kids who seroconvert. So there are some organisms that are different. We have their full genome sequenced. So we have full genome sequences of all of these bacteria. We do not have RNA sequencing from them. We do have metabolite analysis, and we can see that there are metabolites that they produce that are different. And from the full bacterial genomes, we can compare those genomes and ask about or infer genetic capabilities that distinguish the different consortia. So we do have some reasonable molecular handles on what's different about them.
And has it been difficult to identify and culture all these different bacterial strains? Like, I imagine some of them are easier to culture than others. Is that something that could be biasing the results that you get, or the bacteria that you actually get to analyze?
Sure. I mean, we thought a lot about this. That initial archive of about 600 strains that I mentioned to you earlier on that we prepared from these children's fecal samples, they represent somewhere between 70% and 90% of the bacterial complexity of the original fecal samples.
Yeah.
So they're a reasonable proxy, we would argue, for the actual fecal samples, remembering, of course, that bacterial signals that you see in a fecal sample aren't necessarily bacteria that live in that kid. They could also be bacteria that are coming from foodstuffs, et cetera. I mean, the probiotics that we take in from yogurts and so forth, they're certainly present in the fecal samples of people who consume those products, but they're not really part of the community, right? They're dead.
Are you doing just straight sequencing of the stool and identifying, or are you?
Oh, yeah, we had straight sequencing of the stool samples that we worked from, as well as sequencing of all of the organisms that we isolated. So, the organisms that we have are a reasonable facsimile of the complexity of the bacteria that were in the fecal samples. But the advantage of the way that we did them twofold, like all of these organisms grow. They're all alive, right? They were all alive when we isolated them by definition, and they're reanimated when we scrape them off plates and make suspensions and prepare them for introduction into the mice. So we weren't particularly interested in organisms that weren't alive in the samples we had, because obviously they can't be cultured. But yeah, the culturing is demanding, right? It's a labor of love. You have to be willing to work, be able to work in completely anaerobic conditions, because these are anaerobes. They're all obligatory anaerobes. There's some that are facultative anaerobes that can grow in oxygen containing conditions, but we did all of our isolations in anaerobic conditions, and we used more than a dozen different types of media, right? So our goal was to be able to grow the greatest complexity of organisms we could.
So I guess this is a good time to ask, the microbiome we know must vary a lot between patients, but also geographically speaking, we see some of the increases of the diabetes incidents are not necessarily universal in every country or in every region. What can you tell us about geographical differences and how these might relate to the diversity microbiome that we see across the world?
I think these are really fascinating questions, and they were kind of embedded in the point I made earlier about health equity issues. The vast majority of microbiome studies isolation of microbes from healthy humans, et cetera, have really been focused so far primarily on individuals of European origins. And indeed, traditionally, or let's say 50 years ago, the frequency of type 1 diabetes, multiple sclerosis, a variety of different autoimmune diseases was higher in persons of European origin, but there probably was also that appreciation was due to the fact that we really didn't have nearly as comprehensive data from populations living in other parts of the world, Southern Hemisphere, for example, the developing world, as countries that are don't have as much in the way of health research resources. So for low and middle income countries, I think there really is yet a paucity of data on the microbiome, as there was until recently, really a paucity of data on human genetic variations that are more prevalent, or that may be more prevalent in other parts of the world. So this is a challenge to all of us, whether we're interested in human genetics, microbiome diversity, et cetera, that we need to address the need for greater diversity of populations that are enrolled in all of these studies. And it's an enormous challenge. I'm not making light of it. I've been engaged in some of these efforts. I think that there are many researchers, many institutions, many societies of scientists that are really focused on this area now, because it's so vital for health equity. If we find, just trying to put the real silver lining in now, I mean, if we were to find and be able to validate that there are certain compositions of bacteria or certain functionalities of bacteria that are robustly associated with protection from type 1 diabetes, that's the kind of information that you'd like to be able to begin to advance into a therapeutic approach. And to advance therapies that are useful across the world, we have to have cooperating data from many different populations. So this is a key issue. And honestly, if there is one sort of major additional contribution that I could make in the course of my career, it would be to contribute to efforts to bring in a greater diversity of humans into all of these studies of autoimmune disease.
Yeah, there's so much to learn from the diversity, and wouldn't it be nice if you can find this magical combination of probiotics you can give children all around the world for a low price that could reduce the chances of developing this kind of diseases would be very nice.
But would it be a probiotic, or would it have to be a pharmaceutical with pharmaceutical grade manufacturing, pharmaceutical grade safety for prevention of the disease? And then you're talking a drug, and it's not cheap.
Well, that's, I mean, you are the expert, but I mean, that is a problem. That is a problem. Everything becomes so expensive, and then only rich Americans can afford it, or rich European countries.
I think that it is possible to make microbial therapeutics that are not anywhere near as expensive as the cell-based therapies that you may be comparing to in your mind. Sort of like living therapies, right? Living therapies as mammalian cells, like CAR T cells, and living therapies as bacteria. The cost of growing bacteria is a fraction of the cost of preparing mammalian cells. The other piece on which you touch is what are the regulatory authorities going to require in different jurisdictions, in Europe and North America. Maybe put the U.S. to the side just for the moment, because the whole scientific and drug evaluation process is in such a period of flux right now. But let's just say, in Europe and India and other countries, what is going to be acceptable as therapies for little children who aren't sick yet.
Yeah.
Right. I mean, there is no higher bar than that. Yeah. And I think that's going to be a big challenge. But one thing that gives me hope here is that there have been some really spectacular clinical trials outcomes in the prevention of type 1 diabetes using the monoclonal antibody teplizumab or TZELD, which is a monoclonal antibody that detects a invariant component of the T cell antigen receptor.
Mm-hmm.
And this antibody was developed by a small biotech company in California, has now been bought by a very large pharmaceutical company, Sanofi. And that purchase was predicated on some really outstanding clinical trial results where individuals who had these islet autoantibodies that we mentioned earlier, but had not yet progressed to clinical diagnosis of diabetes, were treated in clinical trials with this antibody, and they achieved really remarkable delay or prevention in progression to stage 3. And that's a monoclonal antibody being given to individuals who aren't sick yet. And that may turn out to be the precedent, right, in terms of healthcare providers and regulatory approvals and, all of those pieces.
And it has a companion diagnostic with it to say, hey, you have to have this antibody, which is key, right? They're not just giving it to every kid isn't getting an antibody.
No, there isn't. Well, yes. These are, at the moment, they're giving them to kids who are so-called, or to persons who are seroconverted.
Probiotic or bacterial therapies, if they're derived from humans to begin with, generally in the development side, the tox package and the safety package is much lower because they're commensals, right? And they're oral commensals are surrounded by all the time. So there's still some work to do, but it's not nearly the same as a regular drug, right? Because people are exposed to it all the time, everywhere, all at once. And you're just upping.
I think it will require full genome sequences, however, of all of these bacteria, so that you can credibly argue that you're not transferring organisms that have obviously antibiotic resistance, but also certain kinds of virulence factors. Yeah, because they're very permissive with transferring genes back and forth, right? Once they get in an ecosystem. But yeah, it's going to be a fascinating thing to see.
So I'm glad that you bring up these the therapy for I forgot the name of the antibody. Because I was very curious to know how that works, because I understand it's an anti-CD3 antibody, something like that.
It is, yes.
So it feels a bit. Could you let me know what is the mechanism of action? Are you depleting T cells? Because I guess it's a non-stimulating CD3 antibody.
It was designed to be a non-depleting antibody. The goal was not to create T cell deficiency in persons with diabetes, because you could argue you might create diseases that are less treatable than the one you're trying to already treat or prevent in this case. So it is a non-depleting antibody. The best data from the human trial studies and also some mouse modeling suggest that the antibody in some way converts the physiological state of the so-called CD8 positive T cells, the cytotoxic T cells, and renders a proportion of them somewhat exhausted. So these markers of exhaustion were really defined in models of mice, and then eventually in humans, who were sustaining chronic viral infection. So when there's chronic stimulation of populations of T cells, you can see in CD8 T cells the upregulation of markers on the cell surface that are associated with them now becoming quite refractory to response or exhausted. So that there is evidence in the clinical trials with this teplizumab antibody, the anti-CD3 antibody, that a proportion of CD8 T cells start to express these markers of exhaustion. So that's one thing that could be a mechanism. In mouse models, which were so-called humanized, so they expressed the humanized version of CD3, the target for the antibody, and these also happen to be humanized versions of those non-obese diabetic mouse strain, the autoimmune diabetes prone strain. When they treated under those circumstances, and you were able to actually track where the T cells go, the T cells in that model go to the gut. So they could see the trafficking of T cells from other sites in the mice to the gut. They don't die there, they probably can recirculate other places, and they then became more refractory to immune stimulation. So this may be what's going on in the humans, that is the trafficking to the gut or other mucosal sites, where maybe they're being rendered more quiescent, less reactive. So that's sort of the best data we have now. There's a lot of RNA sequencing that analysis that's going on with peripheral blood T cells being extracted from persons that are going through these clinical trials. Maybe that will shed additional light on the physiological state of these T cells, but they're not being depleted, they're not being killed, but a sufficient number are being reprogrammed, to Jason's earlier point about epigenetics, that they are now expressing programs that are consistent with a lower reactivity, obviously lower reactivity against beta cell antigens too in this case.
All right. So I think we're going to have to wrap it up here. So we'll end with our usual interesting question that's not quite related to science here, which is, what is one hobby you've always wanted to pursue but haven't had the chance to?
Likely this comes out of my twin interests in food and microbiology. I'd love to learn how to make artisanal cheeses. I just find this to be a fantastic way to put together, because cheese is really so dependent, and cheese is the culture, right? It's all about the microbes. That's what really determines the flavors and the profiles of the cheeses we love around the world. I'd love to learn how to make artisanal cheese. Yes.
I love that.
Oh, that sounds really nice.
Yeah, I know. It makes us hungry. Yeah.
Yeah. All right. So it was a really interesting conversation. Also, yeah, I was just thinking that the type 1 diabetes in Toronto has a long history, the first place where children were treated with insulin, actually. So
Absolutely.
now it's a place with history.
Yes.
So we don't have time to go there, but thank you so much, Professor Jayne Danska from the University of Toronto and the Hospital of Sick Children Research Institute in Canada. It was a great conversation. Thank you for joining the Immunology Podcast today.
Great to meet you both. Thank you so much.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on X at @immunopodcast, Blue Sky at immunologypodcast.com, or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.